Thank you, Masood. And thank you for hanging out. So if you heard Jim's talk before about the T-shaped persons, right? I think now we are crawling up the T stem, and we are going on now on the broader picture, and we are not drilling so deep in my talk. So basically, what I want to tell you about is really a little bit from a broader perspective, what are the challenges of modern neuroscience, and what are the open engineering and computer science questions that can actually be now addressed, given that we have a lot of online resources already available and some data out there that anyone who has the skills can actually address and use. So I want to basically take you here in this third point through a brief tour of open problems that actually have some data online and some resources online that can be used. And so if you think about the most challenging science questions, there was 5 years ago in the Science magazine, there was an overview, and they tried to collect the 25 most important science questions. You have some questions that really have to do with where are we coming from, what's the universe made of, and so on. But many of the questions, actually all the ones that are colored now here, are questions that somehow have to do with biology. And as we get here to the red and magenta colors, you actually see questions that have to do with neuroscience, and in particular how our memory is stored in the brain, what is the biological basis of consciousness. Then there's also questions about computing. What are the limits of computing, and how will big picture emerge from a sea of data, of experimental data? And this is in particular something we are facing in neuroscience. So to give you just a brief overview, and I will use what Bruno told you, but to somehow embed this in the broader picture of the brain. Bruno was telling you about basically neural networks on this level. So here you see actually single neurons stained, and this is actually a picture, but it's a drawing, but it is made by Ramon y Cajal, who was looking through the microscope in the early 1900s and basically drew these pictures of neurons where you see that thicker ends are cell bodies, and then they have extensions, dendrites, which is more the receiving end of a neuron, and you will see axons, which are protrusions that actually carry the output signal of the neuron. And so basically these neurons, they are on the size of hundreds of microns. If you go up, you will, so this is the entire brain, and in the colored regions here correspond to systems. So there are different regions in the brain, like the back of the brain, where you actually have visual cortex. So this is something devoted to vision, to the modality of vision. And then you have all the other modalities, and this is basically systems and resides more on the scale of 10 centimeters. And what you have in between is you have within the visual system, you have different maps of visual features. And you have low-level vision and high-level visual areas. And this is basically then on the scale of 1 centimeter. And so you can see that on every level of description, you have structure in the brain. And if you go down, actually, how do neurons communicate? Well, they communicate through synapses. And this here is a micrograph. And you cannot figure out here very much, but when I tell you, so this part here is basically a dendrite of a neuron, and this part here is an axon of another neuron. And this dark line here is basically a synaptic contact. And this is more on the scale of 1 micrometer. You have to use electron microscopy to actually to visualize this. And so you can see that you can basically, to describe the function, for example, how neurons communicate, well, through synapses. So you cannot just limit yourself to one level of description. The challenge in neuroscience is basically to bring together now different levels of description. because neurons interact through something which is much smaller and is almost on the molecular scale, and molecular dynamics becomes important here, and so on. And Bruno already talked about recording techniques. This is just to add here, you have basically these multi-electrode recordings that can record networks and single neurons. But then you have also finer methods where you can actually, so this is a glass pipette, you can patch onto a single neuron and you can record the voltage in the inner of one single cell. And on the other hand, you have these more macroscopic recordings. So these are EEG electrodes, and these are MRI scanner that you can use for structural imaging of the brain, but also functional imaging of the brain. And I will get into that later. So neuroscience is relatively young, but still it's about 150 years old. And it really started out with people like Hermann von Helmholtz, who started to untangle basically the properties of our perception. What do we perceive visually? How can we actually fool our visual sense, and so on? And so this was research that didn't look into the brain, but it studied minutiously, basically regularities in our sensing and perceiving the world. And then people like Ramon y Cajal produced these wonderful pictures of the neurons. So he looked through the microscope, found out that there are different cells, and they interact, and they form fibers that go from one brain structure to the other. And there's beautiful work. His drawings are still one of the most reproduced drawings in neuroscience to somehow demonstrate certain structural properties of the brain. And then you have here people like Hubel and Wiesel, who actually were the first to record with implanted electrodes from neurons and that are visually activated and somehow characterize their response properties. And so those efforts have all in common that they are kind of hypothesis-driven, and that they are focused on one level of description, which is here the cellular level, and here it's the macroscopic behavior level, and that they are all produced basically in one single lab and often by one single investigator. And the results were basically described in words, so pictures in the case of Ramon y Cajal, and they were printed in books or journals. And this was the way of communicating and performing neuroscience. And as Bruno already mentioned, what we have not really yet understood is what do all those neurons do in concert. And in order to understand that, we need to mine these very rich data sets that we are getting nowadays with improved recording technology. So we have to add data-driven approaches to the hypothesis-driven research. We basically have to create infrastructures that allow many investigators to collaborate in mining data sets and producing and complementing each other in producing data sets. And the aim has to be to combine and integrate different description levels, basically. I mean, in particular, the microscopic versus the mesoscopic and so on. And then also publication culture has to change and is changing, so it's now online. It is in journals and in words and pictures, but in addition, it will be very important that also the raw data are accessible for further analysis and meta-analysis. So now, in the remainder of the talk, I will just walk you through some gigantic projects that are currently going on and that all involve their very heavy challenges in terms of ICT. And so the first one is the so-called Microconnectome project, and this is basically the question: if you produce with the electron microscope pictures of thin layers in one small part of the brain, can you somehow reconstruct the full connectivity of such a neural network? And what this implies is basically take these pictures, segment the pictures, so someone somehow has to figure out what is a dendrite and what is an axon, what is a synapse. First on basically a layer by layer level, and then reconstruct in a 3D volume the shape of the neuron and the position of the synapses. And so this that looks like a pile of Persian rugs, basically. It's just such a pile of EM pictures that allow you to do such a reconstruction if you can actually solve this 3D reconstruction problem and before the segmentation problem. It's ongoing work. People are not there. It's a very hard problem, as it turns out, and this is because the data sets are huge. So we are talking here at least terabytes of data for small volumes of small parts of a brain. And but there are some researchers that actually develop these high throughput EM data imaging procedures, and so the data is getting out now and basically is ready also for people who have knowledge in segmentation and 3D reconstruction to actually work on these problems. The second problem, the second big development is the so-called macroconnectome, and so this here you see basically one coronal section of an entire brain. What you can see here, there are these more pinkish there's this outer rim, which is the gray matter, and this is where the neurons are. This is the section that Bruno showed you before, where you see cell bodies. And then you have this lighter part here, which is the white matter. And in the white matter consists only of fibers. It's so to say, it's the information highway of the brain. This is how brain regions exchange information. And the big question is, where do these fibers that go through the white matter, where do they originate and where do they end? In other words, what is the connectivity scheme between these macroscopic regions in the brain? And for animals, we have some very invasive methods to actually trace this connectivity, but not for humans. And so the big project which is now underway is the Human Connectome project. It's a $30 million NIH consortium, basically. And the idea is to try to get connectivity data for humans, for normal subjects, not for patients, just for subjects. And there are basically two different ways how to get at the connectivity data. One uses MRI data, and you can basically image the white matter parts of the brain, and you can actually measure a diffusion, the diffusion of water, which can be and is usually anisotropic. And the elongation of this diffusion tensor is oriented with the typical fiber direction. So in other words, you can use such a tensor image, which is basically a vector field, and you can try to trace the neighboring vectors and basically form a line like here, and this then corresponds to basically the direction of the fiber that you're seeing here. And so this potentially gives you now the ability to tell that regions that lie here in the gray matter actually are connected via fibers that run here through the white matter to areas that lie here in the gray matter. And so this is basically this is one important handle that we have to somehow map the macroscopic connectivity scheme in the human brain. And so the idea, this consortium now basically images a large amount of subjects and puts the data out on the internet. And the big problem, which is still not fully solved, is how do you do this structography? How do you actually, if someone gives you the diffusion tensor data, how do you actually reconstruct these fibers? It's a very difficult problem with local minima, and depending on where you start, you end up with different results and so on. So this is a challenging problem, which is basically still out there for people who have the methods to improve actually the results that they get. And I will be brief. Another way to get the connectivity activity is basically putting people in an MRI scanner. And usually you do that, you tell them to look at something, and then you will see that their visual cortex lights up, and you can somehow trace this functionally induced activity. But you can also lie some subject in the scanner without any task. And this is exactly what people are doing here now. And this sounds silly, but it turns out that you see typical activity patterns. You will find typical areas that in common have higher activity are here in the red spectrum. And there are other patterns that are in the green that are more on the low activity side. And it turns out that these are very stable patterns. You can use different techniques to image them, and they tell something about the individual connectivity of brains. And this is basically the second window to the connectivity structure that we have to the human brain in living subjects and patients. And so this is all, it just started last year, but over the next 2 years, large amounts of data will come out and will be immediately available for analysis on the web. This is the Human Connectome project. And then there are other repositories, web-based repositories out there. For example, those who actually have data how individual cells are formed. And so you cannot see it very well, but there is some green parts which is kind of the dendritic part of the neuron, and the black is the axon, the output side. And so basically, this is a large database where you have the exact morphology of a neuron, and you can use that to, for example, improve the classification of different neuron types. So there's still open questions. And another very valuable thing which can be done with this data is actually to use this morphological shape and actually put it in a detailed neuron model that uses finite element techniques to actually predict function from the structure of the neuron. So this is another example of data which is actually available online. It's a database. And finally, you can also, there's some databases that allow you to actually develop models for predicting neural activity in response to stimuli. And this is, of course, these are neurons recorded in a sensory system, like in the visual system. So, for example, this is here a movie that was shown. And these are here 3 traces. These are now intracellular recordings. These are 3 traces when you repeat the same movie again. And you can see there is variability across these three trials, but there are certain points in time when the neuron tends to respond, and then there are other times when it does not respond. And basically you can now ask, how well can I predict the neural response if someone tells me the stimulus? And there's basically a website actually by our colleagues here, Friedrich Sommer and Jack Gallant, and it's here called the Neural Prediction Challenge. And basically they have here, so this gives you a scoreboard for one of their data sets where people now submit entries, and these numbers here tell you basically the cross correlation between the prediction and the actual model. So the nearer this goes to 1, the better is the prediction. And you can see that over the years, there was quite a steep increase in the ability to predict the model. So people are submitting, every so often they are submitting new models to improve somehow the score. And I will be very brief on the spike sorting. Bruno already introduced to the problem. Basically, if you record from an implanted electrode, you have many neurons in the vicinity. And what you will see just in the electric signal you record is a mixture. And usually people basically just use cluster analysis of the short time kind of signal shapes of these different impulses. And they basically just use cluster methods in order to find out which peak here belonged to which spike, to which neuron. But Bruno basically suggested a better way to do that by using sparse mixture models, that rather than always assigning a peak to just exactly one neuron also allow for the possibility of superpositions by two neurons that spike very close in time with each other. And in a data repository that we run here at the Redwood Center, crcns.org, among many other data sets, there is actually one benchmark data set which is very suited and now basically the main benchmark for testing spike sorting algorithm, because this is a data set where the experimenters not only recorded with the extracellular electrode here, but in addition they basically used whole cell patch clamp recording on some of the vicinity cells. So you have basically a ground truth for that. And just to finish it up, what can I find where? It's confusing. Neuroscience is a huge field. The NIH has actually put out a resource which is kind of a general registry for all kind of neuroscience resources, and it's called the Neural Information Framework, NIF. This is a good starting point. I also didn't talk about data which are actually on the microarray, on the gene expression level, basically also so neuroscience data on the lower level, the almost molecular level that I didn't talk about. This is the Allen Brain Atlas, so they have a lot of data online. And then in addition, there's also the need to basically share tools so that we don't have to reinvent the wheel if we analyze data of a similar structure. And what now emerges is actually Python as a very good language for doing that, because it's open source and kind of non-proprietary. And so a very good, basically a web page that points you to resources for Python in neuroscience you find actually on Fernando Perez's website, who is also a researcher here at UC Berkeley. And also there is actually a conference coming up about this topic, Python in neuroscience. So to sum up, multidisciplinary efforts are somehow required to understand brains. That's pretty clear. Engineering and computer science approaches are very critical for solving this puzzle. And the collaborative online infrastructure is actually now emerging in some points already in place that you can actually join this quest with your laptop if you're interested in the problem. Thanks a lot.
Thank you so much. So we have time for a few questions.
Yeah.
What is the typical fan in and fan out of a neuron?
It depends. In cortex, you have 1,000 to 10,000. The fan in is about 1,000 to 10,000, and the fan out is not much different.
So this is strikingly different from what we do with electrical circuits.
It is, yeah. But this is also, if you look in subcortical circuits, there it's more, the fan out, the fan in is very moderate. It can be 1 to 7 or something like that. The first stage after the eye, for example, where the visual signal passes through, there you have a moderate fan in. But in cortex, it is huge. Therefore, some anatomists call the cortex an information mixing machine.
And are people, after you do the image segmentation and you try to reconstruct the 3D, the connectome, is that being modeled as a graph?
Yeah. Then as soon as you did the reconstruction, you have a graph, you have basically a connectivity matrix, you can apply graph theoretic tools on it and everything. That's one of the main goals.
Does classical anatomy provide any prior information about variation between normal subjects in the Macroconnectome project? And do you think that variation can be detected with the resting MRI?
I think it can be. There are some work in animals that actually show that this can be detected, individual neuroanatomical variability, because it is huge. So, for example, the size of the primary visual cortex can vary by a factor of 2 between 2 different people. So it's really, and also the sulci are different in different people. So it's really, this is also a very challenging problem if you try to map a neural response in one patient on another. This is a big problem, this variability. And definitely rest state fMRI gets you some of that variability.
Thank you so much. Let me again thanks all the speakers that spent time, and also all of you that staying all day with us. So we are finishing our spring lectures, so we're coming back again on fall. Look for us. Just visit the cse.berkeley.edu website, and also the CITRIS website. Will be actually the first week of every month. We're going to have a lecture series beside the special lectures that will come in. Again, thank you so much for being here.